Hello class, today we'll be talking about chapter 2, chemistry, and then part B is going to be biochemistry. We'll do it all in one shot. Feel free to break this up however you need to. So, why do we care about chemistry and anatomy and physiology? Well, the body is of course composed of chemicals, and physiology is sort of underpinned by the chemical reactions that push it forward. So movement, digestion, pumping of the heart, etc. That's just chemistry. Two different types, really two divisions here, a sort of basic chemistry and then a biochemistry. So biochemistry is just the chemistry of life. So we start off with matter. Matter is just anything with mass and that occupies space, can be seen, smelled, and or felt, and it actually feels the effects of gravity, giving us this concept of weight. So three possible states that are really relevant to us. Of course, there are a lot more states of matter, but solid, liquid, gas. And we need to bring in the concept of energy in this. Energy is the capacity to do work. Energy is a massless unit. It does not take up space. And the more work that gets done, the more energy that is used up. It comes in different forms, though. So it can be energy in action, so actually using something to do work, that would be kinetic energy, and it can be stored into a sort of inactive form. And so you can actually interconvert in between these as necessary. Some different forms would include the chemical energy, like those stored in the bonds of chemical substances, electrical energy, such as like passing charged particles through a copper wire, mechanical energy, such as moving something heavy, radiant or electromagnetic energy, like the energy stored in a wavelength, for instance, ultraviolet, X-ray, gamma rays, those have energy. The thing about energy is that you can convert between the various forms. The only downside is it's an inefficient process and you will lose heat as a byproduct of that. That heat is just unusable energy in this instance. So moving on to matter again, matter is composed of the elements, and so the elements are sort of the simplest reducible form of a chemical. And so there are four elements essentially very important for the body, makes up 96% of it: carbon, oxygen, hydrogen, and nitrogen. And so these are largely going to be associated with our macromolecules. Of course, oxygen and hydrogen are in water. Then you have another 9 elements making up 3.9% of the body, and 11 elements making up less than 0.1%. And so you can find these on the periodic table of elements. So the elements themselves are composed of atoms. These are what we would call the building blocks, and the smallest particle of an element with the properties of that element. And so we represent these with an atomic symbol. It's just one or two letters that give the shorthand for that element, so O for oxygen, etc. Some of these may seem unusual, but it's because they come from the Latin names. So Na, natrium, is sodium. Pb, plumbium, is lead. They're just the Latin names. So here's some good figures to review on your own, just showing you the breakdown of how the elements actually work in the human body. But moving on, the atoms themselves are composed of subatomic particles. And it's actually the ratio of these subatomic particles that is giving the atoms their chemical characteristics. But just in general, protons are positively charged. They weigh 1 atomic mass unit. That's just arbitrary. We assigned it. Neutrons have no electrical charge, but also weigh 1 atomic mass unit. Electrons carry a negative charge and are so tiny they virtually have no weight. They do actually, but we tend to disregard that for calculations. So elements are electrically neutral. So that means the protons and electrons will be balanced. So what changes then is the number of the protons and the number of neutrons. The electrons, of course, will match the protons, but the protons and neutrons are found locally in the nucleus, while the electrons orbit around it. And so there are a couple models for how that works. The planetary model is an older, simpler model where the electrons are simply in a path around the nucleus. In an orbital model, you can see that the electrons are actually forming a cloud sort of probabilities. There's a statistical chance that the electron will be found here, instead of right here where you discretely know. So the different elements contain different numbers then of these subatomic particles. And so some basic vocab here: atomic number, mass number, isotopes, and atomic weight. These are all going to be due to the number of subatomic particles. So the atomic number then is the number of protons in the nucleus, and you can see this in the shorthand as a subscript written to the left and to the bottom. The mass number then is the total number of protons and neutrons in the nucleus. So it is actually the total mass of the atom, because we're disregarding the electrons. And so it is written as a superscript to the left of the atomic symbol. So lithium, 3 protons, 4 neutrons. So you'd see atomic number 3, mass number 7. Isotopes. So isotopes are the same elements, so the same number of protons. However, for whatever reason, their number of neutrons actually differ. And so this means that their mass numbers differ. And that might not sound like a big deal, but it can lead to different chemical properties. And it certainly leads to different masses. And so the atomic weight then, which would be reported on a chemical periodic table, is just the average of all the mass numbers of all the isotopes. So some of these isotopes are not stable. They are too large, perhaps. They're interfering with each other somehow. But the atom will actually start to emit subatomic particles as a result of this. And so the isotope will actually begin to decay into a different element. And so as it decays, that particle will release energy. And we call this energy radioactivity. And you can actually measure this. There are several different kinds depending on what type of emission you're actually seeing, but it is due to the decay of these isotopes. And we have lots of application for these. We can use them in imaging, for instance, measuring the emission of these particles as they pass through tissue. Radioactivity can actually damage living tissues, so we can use that to destroy localized cancers, but radiation also causes cancer due to DNA damage. So it is a mixed bag, but we can take advantage of it. So moving into matter and its higher combinations, you have molecules and compounds. A molecule is 2 or more atoms bonded together, while a compound is 2 or more different kinds of atoms bonded together. So a molecule can be the same, but a compound needs to be different kinds. And in truth, most matter is out there as a mixture. And so it's 2 or more components physically intermixed, the 3 basic types of which would be solutions, colloids, and suspensions. So solutions are homogeneous. So the particles actually went into solution and dissolved very evenly. They were dissolved by the solvent, the substance present in the greatest amount. For us, the universal solvent is water, but there are some other pretty good choices. The solute will be the substance actually dissolved in the solvent, and so it's going to be present in smaller amounts and actually go into solution. True solutions are usually transparent. You should not be able to see the solutes at all. So there are a couple ways to express concentration of these solutions. The percent of solute in total solution, so how many parts of solute in 100 parts total. You can measure it in milligrams per deciliter, so mass per volume. And then there's molarity, which is another way of saying solute per liter of solvent of water in number of moles per liter of solvent. And a mole is just a number of particles, actually equal to Avogadro's number. We use that to express concentration. Moving on to colloids. Colloids are also known as emulsions. They are heterogeneous, so they do not mix as well as a solution. Basically, the particles are not evenly distributed. You may even be able to see large particles in solution, and they're not going to settle out. So as a result, the solution will have a cloudy or milky look. And some of these undergo solution to gel transformations. For example, Jello, which is a colloid, goes from a liquid when you pour it to a gel when it sets. Suspensions. So suspensions are moving up this sort of spectrum. They're heterogeneous mixtures, large visible solutes, and those solutes will settle out. So water and sand. If you shake up a bottle of water and sand and let it sit, the sand's going to settle out. Blood, for instance, is actually a suspension. If you leave blood in a tube, the formed elements will start to fall out. So some differences between mixtures and compounds. Mixtures don't necessarily need chemical bonding. The solute-solvent interaction is not necessarily occurring at that level. Mixtures can be separated by physical means, such as straining or filtering, and mixtures can be heterogeneous or homogeneous. Compounds are purely homogeneous. So we've brought in this idea of chemical bonding. So what does that mean? Well, bonds are essentially relationships between chemicals sharing electrons. They're not actually physical structures, they're sharing these sort of statistical distributions of electrons. And so they determine whether a chemical reaction will take place, what type of chemical bond is formed, based on the propensity of that chemical element to actually share. So electrons are occupying electron shells, essentially. These are energy levels. And so depending on its size, atoms have up to 7 electron shares. And due to something called the octet rule, we generally want an even number of 8 in our last shell. Shell 1 only holds 2, shell 2 holds a maximum of 8, shell 3 holds a maximum of 18. The valence shell is the shell with the most potential energy because it's also the furthest from the nucleus. And so those are the valence electrons are the ones that are going to be shared. But atoms want 8 electrons in their valence shell, with the exceptions of the very small stuff. But we call it the octet rule, and it's the driving force behind chemical reactions. One exception to this is the noble gases. This is going to be the sort of furthest right side of the periodic table. Those already have full 8 valence electrons, and so we call those inert gases because they're not going to react. They're not chemically reactive. But in truth, most atoms do not have a full valence shell, and so they will want to trade and interact with other elements until they can get it. So the ability to trade electrons is due to sort of this inherent phenomenon of electronegativity, and this is where we get our bond strengths from. So we have 3 types of bonds here of various strengths: ionic bonds, covalent bonds, and hydrogen bonds. Ionic bonds, whenever they lose their electrons, they'll become charged. And so then the number of protons is not equal to the number of electrons, and due to this transfer, you have one becoming negative and one becoming positive. So if it's an anion, it will have a negative charge. If it's a cation, it will have a positive charge. And of course, opposite charges attract. So ionic compounds, a sort of union between these anions and cations, will bind together, and this is where we get our salts, for instance. So NaCl, sodium chloride, one of them is going to be an anion, one of them is going to be a cation. But that is actually an ionic bond. And so even though we represent it as this NaCl, in truth it's just a ratio, and the actual form out in the environment is going to take the place of something like this: large numbers of these sodiums and chlorides working together. So what about covalent bonds? Well, covalent bonds are sharing of 2 or more of the valence shell electrons. You're not actually changing the electrical state like you were in the ionic bond. And this is where we get our single, double, and triple bonds, depending on whether you shall share 2, 4, or 6. And so because of the sharing here, each atom gets to fill its valence shell at least part of the time. And so there's two types of covalent bonds, polar and nonpolar. So nonpolar covalent bonds share their electrons equally. That's why there are no poles. The electron, you can just imagine, is just moving around each atom around the same amount of time. And so it is electrically balanced, and that would be something like CO2. So linear, symmetrical, nonpolar, electrons being shared very evenly. If a covalent bond is polar, then there is unequal sharing of the electrons. And so this is an electrically polar molecule and has electron attracting, different atoms have different electron attracting abilities. We call that electronegativity. And so, for instance, H2O is a polar molecule because the electronegative oxygen is pulling on that shared electron from those hydrogens, creating, in this case, a dipole. But you could also have like a monopole, in which case one atom is just hogging the electron. So let's talk about hydrogen bonds. So really, this is going to be more of an interaction than a bond, I would say, but it is an attractive force between adjacent hydrogens of one molecule to another. Not true bonds, like I said, more of an attraction. But these are very important because of the way our macromolecules interact. There's a lot of hydrogen bonding characteristics there. But this is also what makes water liquid. So it serves as intramolecular bonds holding large molecules in their three-dimensional shapes. Okay, moving on to chemical reactions. So matter cannot be created or destroyed. And so when you look at chemical processes, there's always going to be a reactant side and a product side, and those have to be able to match up. And so chemical reaction is simply when chemical bonds are formed or arranged or broken to produce product side and reactant side. We represent this as an equation with reactants and products, and then we balance those out using an equation. You can represent these complex formulas with a molecular formula. For example, H2O water, C6H12O6 would be glucose. The subscript is going to indicate the atom joined by the bonds, and the prefix will tell you how many unjoined atoms or molecules you have. So types of chemical reactions, you have synthesis reactions where smaller particles are bonded together to form larger particles. You have decomposition reactions where a substance is broken down into smaller molecules. That would be catabolic in nature, so bond breaking. Like that. So you can have exchange or displacement reactions where synthesis and decomposition are both taking place. You can have redox reactions where one side is getting reduced, one side is getting oxidized. That is due to the loss or gain of electrons. And all of these are either taking energy or releasing energy. So an exergonic reaction results in a net release of energy. It's giving off energy, and the products have less potential energy than the reactants. Endergonic reactions result in a net absorption of energy, and the products have more energy than the reactants. Have more potential energy, sorry. So the exergonic would be catabolic, and endergonic would be anabolic. Think about the energy that goes into forming a bond, for instance. All chemical reactions are theoretically reversible. Due to the energy states of product and reactant, it's not always feasible to do that, but many biological reactions, for instance, have to be catalyzed due to the energy requirements, so that would require the help of an enzyme. But things that affect speed of a chemical reaction, temperature, concentration of the reactants, and how big is the particle size. So I mentioned the idea of catalyzing a reaction. What does that mean? Well, a catalyst is increasing the rate of the reaction without actually changing the energy levels of the products or the reactants. Instead, what it's doing is lowering the activation energy. And so enzymes are our biological catalysts. 99% of the time, they're amino acid based proteins usually. About 1% of the time, or maybe a little less, they're RNA based. Ribozymes. Okay, so that was part A, chemistry. Now this is part B, biochemistry. There's going to be some common themes, some common iterations in our macromolecules as we go through, but start us off. Biochem. It's just a portmanteau of biology and chemistry, so it's exactly what it sounds like. It's the study of living matter. And so all chemicals are either organic or inorganic. Inorganic compounds are going to be things like water, salts, acids, bases, metals, for instance, are inorganic. But organic compounds are anything with sort of like carbon and hydrogen. So carbohydrates, fats, proteins, and nucleic acids. They are large. They're usually covalently bonded. Carbon loves to covalently bond stuff. And both are important for life. So let's start off with water. Water is the universal solvent. It's the most abundant inorganic compound. It's 60 to 80% of the volume of living cells. And it has some characteristics that make it really useful for living systems. So for instance, it has a very high heat capacity. So it takes a lot of energy to change the temperature of water. It's a high heat of vaporization. So for one thing, it doesn't flash off very fast when it is heated, and when it does, it takes a lot of energy with it. So we can take advantage of that for evaporative cooling. It is polar and in some cases I think protic or aprotic, but it has polar solvent properties, so you can dissolve a lot of solutes in it. It has some reactivity, so the hydrogen bonds can be broken. It can change state. And it has cushioning effects as well. It doesn't compress very well, so it basically absorbs a lot of energy and force. High heat capacity, so resist temperature change. High heat of vaporization provides us a useful cooling mechanism. Polar solvent properties essentially can form large hydration shells around charged molecules like proteins. This gives us a transport medium as well, of course, because you can dissolve stuff in it. Reactivity, so useful in hydrolysis and dehydration synthesis. So as we'll see, these larger molecules are actually made with dehydration synthesis reactions. Cushioning, of course, protecting organs from physical trauma. We surround our organs, certain organs, with fluid cushions. Salts dissolve very readily in water. And they will actually separate out into their cations and anions when they separate out. We call these ions electrolytes. And so these electrolytes are actually responsible for conduction of the electrical currents that we use in our biochemistry. And so examples of that would be sodium, potassium, calcium, and even iron in some instances. And so maintaining this ionic balance is very important for homeostasis. We use these salts for lots of things, for instance, but moving on. Acids and bases. Acids and bases are also electrolytes, and they ionize and dissociate in water. Really, the strongest acid or base that can exist in water is water, essentially. And so it will just simply dissociate these down. In the simplest terms, acids are proton donors. When they go into solution, they will release hydrogen ions, which are essentially bare protons, so a charged subatomic particle. And some important acids will be something like hydrochloric acid, acetic acid, carbonic acid, for instance. Those will come up in the future. Bases, in their simplest form, are proton acceptors. They will accept those hydrogen ions from solution. So when they dissociate, they release hydroxyl ions instead. And some important bases in our body include bicarbonate ion and ammonia. So concentration, we measure with a pH scale. It's a relationship between hydrogen ions in solution and hydroxyl ions in solution. but basically it is a logarithmic scale that goes from 0 to 14. So basically every time you go up 1, it goes up a magnitude. So a tenfold difference. A pH 5 solution is 10 times more acidic than a pH 6 solution. So acid solutions have high hydrogen concentration but low pH. So 0 to 6.99 is acidic. Neutral solutions have equal numbers of the hydrogen ion and hydroxyl ion, so they are pH 7. If you look at pure water, like actual DI water, the pH will be 7. Alkaline or basic solutions have low hydrogen concentration but high pH. So 7.01 to 14 is alkaline or basic. And so here's a good example of that. You can go from 0 with hydrochloric to up here to 14 with sodium hydroxide, and of course in the middle will be DI water. So if you mix an acid and base together, a displacement reaction will occur and they will actually neutralize each other. Now you want to be able to resist changes in pH, and to do that our body actually has buffering systems. So a buffering system will resist any type of rapid change, a swing in pH. Basically, it will release hydrogen if the pH rises and bind hydrogen if the pH falls. And it does this by using a relationship between strong acids and weak bases. Strong acids or bases will completely dissociate into weak ones. Our system is the carbonic acid bicarbonate system, which is an important system of blood, and then there is actually a phosphate buffering system as well. So moving into organic compounds. Organic molecules contain carbon. The exception to that would be CO2, which is the most oxidized form of carbon. That is not organic, that is inorganic. But carbon is electroneutral. It likes to share electrons and never gains or loses them. And it likes to form 4 covalent bonds with other elements. But it will also form 4 with itself as well. Carbon is unique to living systems. Well, I'm not sure where they're going with that. There are lots of minerals carbon in them. But the major organic compounds that we talk about, the macromolecules, are these 4 important classes of molecules that have carbon backbones. So these are polymers. These are chains of units called monomers. All will be synthesized by dehydration synthesis, so using water. And similarly, they can be broken down with water in a hydrolysis reaction. And so we'll start off with the carbs. Carbs include the sugars and starches. They contain a ratio of carbon, hydrogen, and oxygen in a 2-to-1 fashion. You can have monosaccharides. Those are the simplest. Those are the monomers. 1 single sugar. A disaccharide would be 2 sugars bonded together, and then a chain of these would be a polysaccharide. So the simplest sugars contain 3 to 7 carbon atoms, and they are just the monomers of the carbs. Important ones would be the pentose sugars, which of course we use in our nucleic acids, ribose and deoxyribose, and then the hexose sugars, like glucose. So here's those. Disaccharides are the double sugars. So important fact about these is that they're actually too large to pass through the selectively permeable cell membrane, but this would be something like sucrose, which is what our table sugar would be. So polysaccharides are the polymers then of these monosaccharides. Again, they're formed by dehydration synthesis, so they're removing a water to form a bond. Important polysaccharides would include starch, which is the storage form used by plants, and then glycogen, which is the storage form used by animals. Now, we don't have cell walls, animals don't, but when you look at cell walls out in the various kingdoms, they're going to be polysaccharide based. So plants use cellulose, fungi use chitin, for instance. They are carb based, but not very soluble. Moving on to the lipids. Lipids, kind of a strange class. They are not structurally related, but functionally related in that they are insoluble in water, but contain carbon, hydrogen, oxygen, and sometimes phosphorus as well. We're going to see some phospholipids. The main types will be triglycerides, phospholipids, steroids, and eicosanoids. And the pigments are also in here as well. So triglycerides are fats when solid or oils when liquids. They are composed of the fatty acids. In a triglyceride, it is 3 fatty acids bonded to a glycerol molecule. Glycerol is just a sugar alcohol. Its function is energy storage, insulation, and protection. So you can see the glycerol molecule up top, and then one of the fatty acid tails down here at the bottom. So here's one with 3 fatty acid tails. So these can be saturated or unsaturated. If a fat is saturated, all the carbons have single bonds between them, and it has the maximum binding capacity for hydrogen. So it is saturated with hydrogen ions, with atoms. So as a result, these are highly linear, and they can be packed closely together, forming a solid at room temp. So your saturated fats are going to be like animal fats, butter, something that stays solid at room temp. Unsaturated fatty acids, what happens is the carbons will start to double bond and introduce kinks in there. And because of that reduced hydrogen capacity and that kinking, these will not stack well together, and the resulting product will be liquid at room temperature, so something like an oil. Mentioned phospholipids before, so these are modified triglycerides. They glycerol and 2 fatty acids, but now you have a phosphorus containing group where that other fatty acid tail was before. And so the head and the tail regions are going to have different properties. The head will be polar and hydrophilic. If you look at the etymology of the word, water-loving. The tails will be non-polar and water-fearing, hydrophobic. So this is actually what makes up our cell membrane structure. Then you have these steroids. These are a series of rings. The most important steroid is cholesterol because it serves as a precursor to useful stuff for us like bile salts and vitamins, but also is intercalated into the plasma membrane to introduce rigidity and stuff. But steroids, very important, precursor to hormones, but all have this 4-ring structure. Then moving on to the eicosanoids, derived from fatty acids. These include the prostaglandins. These are kind of signaling molecules that control blood clotting, pressure, inflammation, labor contractions. This is where our pain response is coming from. If you take an NSAID, then you're trying to block essentially the prostaglandins. Okay, moving on to proteins. 20 to 30% of the cellular mass. Proteins are kind of ubiquitous because they're the functional elements of the cell. They're where the work is getting done. And so they have the most varied functions of any molecules because they have the most jobs. So it can be structural, can be chemical, they can be in motility, it can be storage, but contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur, phosphorus I'm less sure about, but can also contain selenium. So the polymers of amino acid monomers are held together by peptide bonds. Peptide bonds link adjacent amino acids. These are created by dehydration synthesis, just like we saw before. And there are 4 structural levels that we really talk about. I guess they'll come in the future, but 4 levels of organization here. So all proteins are made from 20 types of amino acids. In truth, we have many more types. There are more than 20 amino acids. We learned 20, but joined by covalent bonds called the peptide bonds. You can tell an amino acid because it has an amine group on one side and a carboxylic acid group on the other. And so it can act as either an acid or a base. And all amino acids have that characteristic. What changes the amino acid is which of the 20 different R groups is present. And so you can see here the amine group, the acid group. This side is basic, this side is acidic. The only thing that changes is the R group here. And they can be simple. It could be as simple as a hydrogen atom. They could be quite complex. So I mentioned before there are structural levels to the protein, and this is important in how we study them as well. The first will be the primary, and so primary structure of a protein is just the linear sequence of amino acids. If you laid this out like you were writing it out on a piece of paper, what would the order of the amino acids be? That's the primary structure. That actually determines the shape and the function. The secondary structure would be due to hydrogen binding between these sort of localized R groups. So how primary amino acids interact with each other, mostly through hydrogen bonding. And there are two forms that this takes, predominantly. The alpha helix, which will coil and resemble a spring, and the beta pleated sheet, which resembles ribbons. And so alpha helices provide flexibility, beta pleated sheets stack together very nicely and provide rigidity. Then there's also tertiary structures. So how are our secondary structures interacting, and what is the overall form of our protein? And then finally, this is sort of optional. Not all proteins have quaternary structure, but how do two or more of these different polypeptides interact with each other? Basically, does the protein have subunits? If so, then it has quaternary structure. So protein shapes really fall into two classes. They're either fibrous or globular. If it's fibrous, of course, it's strand-like. It's very stable, kind of water-insoluble. Most have tertiary or quaternary structure, and their use is mechanical support and tensile strength. These are going to be fiber proteins like keratin, elastin, collagen, and contractile fibers. Globular proteins, on the other hand, are compact, they're spherical, they're water-soluble, and they may be sensitive to environmental changes. They can have tertiary quaternary structure as well, but they're going to have specific functional regions called active sites where they're going to interact with the substrate in order to do some kind of task. And so examples of these would be antibodies, the peptide-based hormones, chaperone proteins, which help fold other proteins, as well as enzymes, which of course catalyze reactions. Now, one thing you have to be careful with: proteins are weak to denaturation, essentially, especially these globular proteins. When they start to unfold, when their hydrogens start to deprotonate, they will lose their 3D shape. And when you lose shape, you lose function. And so fibrous proteins are more stable in this way because they're much harder to simply just lose their shape. What happens, though, is your active site becomes deactivated. And so any kind of decreased pH, which is increased acidity, or even increased temperature, is going to cause this denaturation. And it is usually reversible. If normal conditions are restored, the protein will spontaneously reform into that native state. But you cannot undo cooking an egg, right? If changes are too extreme, it will stay like that. Okay, moving on to enzymes. Enzymes, they're saying globular proteins that act as biological catalysts. Guess that's true, but catalysts regulate and increase the speed of chemical reactions without getting used up in the process. Just keep in mind there are other biological catalysts than enzymes, but lower the energy needed to initiate a chemical reaction. As a result of this, the speed of the reaction is going to increase by an incredible amount in some cases. This will allow for millions of reactions per minute. So these enzymes are really necessary in cases where the activation energy is too high for the reaction to proceed spontaneously. So most functional enzymes are going to be referred to as hollow enzymes, have two parts. The apo enzyme is the actual amino acid component, and then they'll need some type of activator. And so this is where our trace elements actually come in. So our trace elements are vitamins and minerals. They are serving as cofactors in this case metal ions, or coenzymes, which are organic molecules. A cofactor binds an apoenzyme to activate it and make a holoenzyme. These enzymes are very specific and act on a specific substrate. You can tell something is an enzyme if it ends in the ACE suffix, so ASE. And they're often named for the reaction they catalyze. For example, a hydrolase is an enzyme that is catalyzing this hydraulic reaction. Enzyme action. So how do they work? Well, they are changing the activation energy to prime the reaction, allowing the reaction to proceed quickly and at body temp. 3 different steps here. First, the substrate will bind the enzyme active site. The complex will undergo an arrangement resulting in the final product. Then the product is released from the enzyme. So when I say activation energy, this is what I mean. So you can see in part 1 here, without the enzyme, the reaction is not able to proceed until the activation energy is lowered. So here is that mechanism of binding the active site. The enzyme substrate complex is formed, the bond is formed here, and then the product is released. Okay, moving on to our last macromolecule, the nucleic acids, composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus, largest molecules in the body. So the nucleic acid polymers are composed of monomers called nucleotides. A nucleotide is a nitrogenous base, a pentose sugar, and a phosphate. There are 2 major classes, the deoxyribonucleic acids and the ribonucleic acids. These only vary by 1 oxygen. It's just that 1 oxygen has to be important for stability. So DNA is the hereditary molecule. It holds all the genetic information. It is double-stranded typically, so organized into a double helix that is anti-parallel. In eukaryotic cells, that is going to be located in the nucleus. And of course, the nucleotide will have that sugar, which is deoxyribose, a phosphate group, which is giving a slight negative charge, and then one of 4 nitrogenous bases. Of the purines, adenine and guanine; of the pyrimidines, cytosine and thymine. And so, like that. Then you also have RNA. So RNA acts as an intermediate between DNA and proteins, basically telling the protein the DNA's instruction. Typically single-stranded, typically linear. Active outside the nucleus, there are immature RNAs inside the nucleus that need to be sent out and be processed, but contains a ribose sugar, not deoxyribose. And instead of thymine, we're using uracil. We have 3 very important varieties of RNA for DNA to protein synthesis. That would be messenger RNA, transfer RNA, and ribosomal RNA. There are actually many small RNAs, though, that are vitally important to the cell. Another useful nucleic acid will be ATP. This is actually the chemical currency of the cell. We yield it by aerobically processing glucose, and it powers chemical reactions in the cell by providing a usable energy source in the form of an inorganic phosphate that it can transfer. And so it's an adenine-containing RNA nucleotide with 2 additional phosphate groups. So adenine, or ribose sugar, and then 3 of those phosphate groups. What happens then is that one of those terminal phosphate groups of ATP gets transferred, and now it's using that phosphate bond to essentially store work. And ATP synthesis is used to couple essentially inorganic reactions that would not normally be able to apply via this process. So this process, hydrolysis of ATP to ADP, is an energy yielding process, very important for the cell. Does a lot of cellular work that otherwise would not get done just due to the nature of the products and the reactants. Okay, so that was chemistry and biochemistry. Have a good day.